Hello, and thank you for coming to this webinar on real use case examples of Nanolive's live cytotoxicity and live T-cell assays. My name is Hope Amos, and I'm the Scientific Communications Specialist here at Nanolive. We're so excited to share our technology and present some of our first groundbreaking results that our key opinion leaders in industry have been kind enough to allow us to share. As always, during the event, please submit any questions and quote requests into the chat box, and I will reply to you via email after the event. So today, first I will give a brief overview of Nanolive's technology and then jump into the case studies. First, we have a dataset from Lightchain Bioscience analyzed with our live T-cell assay, screening bispecific antibodies on a T-cell target cell co-culture without using fluorescent labels. Then we have 3 datasets analyzed with the live cytotoxicity assay, an investigation into mRNA uptake, expression, and cytotoxic effects carried out by TrON. Then testing selectivity of senolytics in a co-culture by scientists at dsm-firmenich. And finally, how the assay can be used to quantify uptake of particles by cells with a dataset from bit.bio. So let's start with a look at our imaging platforms, which use digital holotomography to produce high-resolution 3D images label-free. The 3D Cell Explorer 96 Focus was newly released this year, and with its laser-based autofocus and 96 well capacity, it enables long-term continuous monitoring of cells in up to 96 wells. Digital holotomography uses the intrinsic refractive index of cells and their components to distinguish between cells, organelles, and other materials, and visualizes their movements without any labels or phototoxicity. Therefore, the value of each experiment is greatly increased thanks to the high-content, high-resolution data we can capture in a single acquisition. Our AI-powered digital assays take this output from label-free data and digitally segment cells to analyze every cell individually and to produce quantitative phenotypic metrics. Nanolive's digital assays enable you to increase the predictive value of in vitro assays by increasing the number of parameters studied, with high-content imaging giving a greater understanding of cellular response to drug candidates. In vitro relevance is improved by quantifying the responses of subpopulations of cells in co-culture. This more accurately represents the in vivo environment. The assay enables you to accelerate drug discovery with automated workflows, multiplexed experiments, and walk-away imaging. They also simplify experimental workflows by reducing the amount of sample prep needed and by standardizing acquisition and analysis to reduce user errors and variation. And finally, measuring cytotoxicity and other phenotypic changes label-free allows the collection of unbiased data. One such phenotypic change that can be measured label-free is cell death, as demonstrated in this video. AI is used to classify cell viability, and dead cells are classified as apoptotic, shown here in pink, or necrotic, which are shown in dark blue. No fluorescence was used to produce these images, and Nanolive is the only platform capable of identifying the type of cell death automatically and label-free. Label-free detection of cell death is included in 2 of our digital assays, the live T-cell assay and the live cytotoxicity assay. The live T-cell assay was specifically designed to quantify the interactions between adherent target cells and T cells in co-culture and to measure target cell death. The live cytotoxicity assay is our first assay with the ability to divide a population of cells in co-culture by their fluorescent signature. This allows trends in cell death rate and type of cell death to be recorded separately for different cell lines in the same dish using only a single fluorophore. The fluorescent signal within every cell is also quantified to allow measure of drug uptake or gene expression levels, as I will show today. The first case study I will cover today was analysed by our live T-cell assay. This digital assay classifies cells as target cells, which you can see here coloured in blue, or T cells, which are coloured in red, and their interactions are shown in yellow. Again, this assay is completely label-free, and the colours you see here have been assigned by the digital assay software. Quantifying interactions provides insight into therapeutic mechanism of action by providing dynamic data on binding, including the number of T cells in contact with target cells at every imaging time point. Label-free detection of target cell death enables study of T cell killing rate for understanding of the effects of therapeutics and efficacy of CAR T cells. Kinetic data over long time periods allow the T cell response to be better understood, detecting spikes in activity or T cell exhaustion. And the live T cell assay is well suited to applications involving patient samples due to the label-free nature of the assay and the fully regulated imaging environment in the stage-top incubator. In the first case study, we will look at how the live T-cell assay was used to screen antibodies to assess their effect on T-cell killing. I will show how with this digital assay, you can measure the death rate of target cells in a co-culture with T-cells label-free, quantify the dynamic T-cell response to therapy, and compare different donor T-cell responses to the same antibodies. We would really like to thank the team of Valerie Moine in the pharmacology unit at Lightchain Bioscience for sharing these fantastic data with us. T cells are heterogeneous, and T cell response to treatment varies from person to person, highlighting the need for more personalized approaches to cancer therapies. This investigation was designed to test the ability to identify the differences in T cell response at the in vitro stage without perturbation of samples by fluorescence. For this experiment, T cells were collected from 2 donors, then added to adherent cancer cells in 96 well plates. First, the antibody or control was added to each well. Then T cell target cell interactions were detected. Successful therapies triggered an increase in target cell death, which we also measured with the assay. First, let's have a look at the acquisition videos where the live T cell analysis has already been carried out. You can see that the T cells and target cells were detected with high accuracy. In the negative control well in the bottom left, you can see there is a higher proportion of red T cells which are not interacting with the blue target cells. In comparison, in the antibody 2 well in the top right, you can see more yellow interactions as T cells bind the target cells and begin killing. And just a quick reminder again that these colours are not fluorescents and they have been digitally assigned by the assay. Of course, these images are very beautiful, but let's have a look at the quantitative data to get some more detailed information. These graphs show the T cell killing dynamics in each well over time. We see that all antibodies increase the amount of target cell death in comparison to the negative control in the bottom left. Furthermore, antibody 2 had a greater cytotoxic effect on target cell death than antibody 1. The faster cell death caused by antibody 2 is shown by the steeper gradient, as well as a larger amount of cell death which occurred over the same time period. The live T cell assay can also quantify the interactions between T cell and target cells to measure things like the percentage of T cells in contact with target cells over time, or the mean distance between T cells and target cells as shown here. This metric gives us an insight into the antibody's mechanism of action. We already know that the antibodies increase target cell death, but here we can see that they each decreased the mean distance between the different types of cells in comparison to the negative control, which is plotted in grey. This likely reflects an increase and change in T cell binding induced by the antibodies. And these are the results of treatment with the positive control for the two different T cell donors. In this case, we can see an early peak in cell death in donor 2, which was not seen in donor 1. This difference wouldn't have been detected if we were just using an endpoint assay, as this captures only a snapshot of the T cell response. And this highlights the importance of using live cell imaging like Nanolive for looking at T cell interactions and T cell response. So to summarize the results of this case study, we have shown that the label-free dynamic data from the live T cell assay can be used to provide high-content data about the T cell response, including target cell death rate and finding and binding dynamics. The versatility of the Nanolive platform's label-free live cell imaging enables faster prep time and reduced use of costly therapeutics and valuable patient samples. As well as screening therapeutics, as I've shown here, the live T cell assay can be used for various immuno-oncology applications, including using the dynamic output to detect T cell exhaustion and to verify the health and function of CAR T cells. The next case studies were analysed with Nanolive's live cytotoxicity assay. This assay has the same label-free detection of cell health and death, combined with the ability to classify cells into subpopulations by their fluorescent signature. On the left of the screen, you can see a co-culture of cells tagged with a fluorescent red marker and the unstained cells, which are visible thanks to the refractive index imaging. The view on the right shows how the software detects the boundaries of these fluorescent cells, and the boundaries are shown in yellow, and this allows the subpopulation to be defined and death metrics to be split by cell type. With the live cytotoxicity assay, you can investigate 2D co-culture microenvironment models to better recapitulate the tumor microenvironment and to detect the influence of non-target cells from the stroma on target cell responses. For uses in drug safety and investigative toxicology applications, you can quantify drug specificity and cytotoxicity on co-cultures of target and non-target cells. Monitoring drug delivery is now possible by visualizing and measuring the uptake of fluorescent drugs or particles. And the live cytotoxicity assay can be used for studying RNA therapeutics by measuring the expression of mRNA and cytotoxicity simultaneously. I will cover case studies for these 3 scenarios next. The first case study involves measuring expression and cytotoxic effect of mRNA treatments. Here we use the live cytotoxicity assay to measure the change in intracellular fluorescence corresponding to mRNA expression and compared this with cell death, which was detected label-free. And a big thank you to Mario Perkovic, head of gene transfer, and his team at TrON, the Center for Translational Oncology in Mainz, for allowing us to share these data today. The experiment tested 4 therapeutic mRNA candidates with 2 negative controls and 4 replicates each. Cells were imaged every hour for 22 hours. First, mRNA was added to cell media and was taken up by cells. A fluorescence reporter was produced by mRNA expression, increasing cellular fluorescence. And both fluorescence intensity increase and cell death were quantified over time by the live cytotoxicity assay. The assay was first used to quantify fluorescence intensity per cell over time. This revealed a difference in the mRNA expression between wells, with RNA4 expressed at the highest levels, followed by RNA2. Cell death was also quantified for each condition, and again, there were differences between the responses across the different wells. So looking at the same data, comparing the fluorescence intensity, or the RNA expression, against cell death in the same well can enable us to contextualize the results. So when we first plot fluorescence intensity, like I've done here for RNA4, we can see a wave corresponding to the increase in RNA expression. This signal rapidly increased and peaked before the 8th hour of imaging. Now, by plotting the cell death fraction on the same graph on the y-axis, we can see the relationship between the RNA expression in green and the cell death in grey. So here we can see that cell death peaked roughly 11 hours after the peak RNA expression. We can perform the same comparison for every well, and this allows us to look at the differences between RNA expression and cell death across all conditions. RNA2 and RNA4 both caused similar levels of cell death, with similar kinetics and time lag between the peak expression and the peak cell death, but RNA2 was expressed at roughly half the level of RNA4. Therefore, we can infer that RNA2 had a stronger cytotoxic effect. RNA1 also caused relatively high cell death relative to the RNA expression, which was relatively low. And RNA3 had both low expression and low cytotoxic effect. These results show that the live cytotoxicity assay can be used to detect mRNA expression levels and cell death kinetics to identify the most and least cytotoxic RNA relative to expression. Measuring both mRNA expression and cell death simultaneously increases the robustness of data, as any variations in transfection efficiency are already accounted for and are reported by the fluorescence intensity measurement. Now I will cover how the live cytotoxicity assay's ability to split co-cultured cells into subpopulations can be used to measure drug selectivity. Senescent cells in skin are a target for anti-aging compounds known as senolytics, which can selectively kill these senescent cells. As cosmetics ingredients cannot be tested on animals, Nanolive's high-content imaging platform can give greater context and translational relevance to in vitro studies. We are very grateful that we could share an example of this targeted killing today in this dataset from the team of Lightchain from dsm-firmenich that produces ingredients for the cosmetic industry. Primary human dermal fibroblasts were co-cultured with senescent cell lines to assess drug specificity. In this co-culture, the senescent cells were treated so that they fluoresced, so that the live cytotoxicity assay could distinguish them from healthy cells. So to give a quick overview of the experiment, first, the senolytic drug was added to the co-culture. It specifically killed the fluorescent senescent cells, and the assay separated cell death for each cell type. By looking at cell death for senescent cells, which was plotted in green, and the healthy cells in grey, we see that indeed the live cytotoxicity assay was able to specifically detect cell death in the different populations. The death kinetics were as expected, with lower cell death rate in the healthy cells. This method could be applied to test the selectivity of other targeted drugs and to detect the bystander effect of antibody drug conjugates. I will use the final case study today to demonstrate how the live cytotoxicity assay can be used to measure changes in fluorescence intensity, and how the refractive index imaging can be used in parallel to understand particle uptake and phagocytosis. This dataset was kindly shared with us by the team at bit.bio, who use a proprietary method to reprogram pluripotent stem cells to a new cell identity, and features their IO microglia cells. bit.bio's IO microglia were co-cultured with pH-rhodo red dimerzan bioparticles, which fluoresce only when exposed to an acidic environment, such as in the phagosome after internalization by cells. Therefore, by measuring intracellular fluorescence over time, the bead uptake can be quantified. So first, non-fluorescent particles were added to the cell media and were taken up by the IO microglia. Once internalized by the cells, the acidic environment of the phagosome triggered fluorescence, which was captured by the Nanolive imaging platform and quantified by the live cytotoxicity assay. This is a snapshot image from the data capture showing the refractive index imaging in grayscale with the red fluorescence overlay. Non-fluorescing particles can be seen in the cell media, indicated on the left close-up by white arrows. You can also see 3 internalized red fluorescent particles inside this cell, indicated by the red arrows. And in the neighboring cell, we can see a particle that has been recently taken up and which has not yet started to fluoresce. Here, the mean intracellular fluorescence per cell was used as an output to quantify the uptake of particles and the resulting increase in fluorescence intensity in cells for the imaging duration. The graph on the left shows us how quickly the particles were taken up by cells and how long it took to reach maximum uptake. So here we can see that the uptake of particles was linear until the maximum was reached at around 1 hour 15, when all particles had been internalized. The refractive index imaging on the right shows cell membrane dynamics and reveals how the particles are engulfed and internalized by the cells. This method enables quantification of fluorescent particle uptake without manual counting or manual image processing. All the segmentation of cells and the fluorescence quantification has been done automatically in the software. So to summarize briefly, in this example, we used fluorescent particles, but the assay could be used in the same way as shown here to monitor the uptake of drug delivery particles like liposomes tagged with fluorescence as well. So to summarize, Nanolive's label-free cell death detection enables the monitoring of cells in unperturbed conditions and the collection of high-content data. This technology is used in the live T-cell assay to provide comprehensive characterization of the dynamic T-cell response. T-cell count, target cell death, and T-cell target cell interactions are just some of the metrics that you can record with this assay. The live cytotoxicity assay combines fluorescence with this label-free analysis to quantify intra and extracellular fluorescence and cell death kinetics in co-culture subpopulations. We would like to end the webinar today by again thanking our clients in industry and academia for choosing to use Nanolive imaging, and a special thank you to the key opinion leaders that shared their data with us today. We wish you all continued success in your research. And finally, thank you to everyone who joined us today. Please reach out to us with any queries or questions. Our team is always happy to help. You can contact us at lookinginsidelife@nanolive.ch or by filling in a contact form on our website. We hope to see you at our next event.